Okay, so today we'll be starting with Unit 3 and we'll be continuing our unit and we'll be moving on to photosynthesis and respiration. So, before knowing anything about photosynthesis and respiration, you first need to learn about electron carriers. Carriers. And they are of mainly two types which we'll discuss in these videos, and that is NADH and FADH2. So what these two molecules do is they basically carry two electrons and they basically deliver it someplace, and delivering it means it's like an input of energy, it's like a medium of energy, and and these carriers are like the transport through which you transport that energy. So for example, if electron is the coal, then these are like the truck who carries it. So now we'll first talk about photosynthesis. So when talking about photosynthesis, the first thing that the first thing that should come to your mind is chloroplast, since photosynthesis happens on a very cellular basis, and that cell is called a chloroplast. And so chloroplast contains of a few 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 features. So we have these pancakes. You might have heard them all while watching or attending a lecture about these. But these are basically like thylakoids, and thylakoids like absorb the energy, and the light independent reaction happens in the stroma. So this is the stroma, the empty space, and then we have the inner membrane, which is like separation of the. This inner membrane comes in handy when we're talking about the proton gradient later on in the light dependent reactions part. So this is basically that, and I think we've covered all we need to for this bio. So only some things are important. So you need to know the thylakoid, you need to know the stroma, inner membrane, and basically one this pancake is a thylakoid, whereas multiple are granum. And you don't have to do lamella in depth right now, but it's like the connecting strand of it. So now, when we're talking about photosynthesis, we have two reaction centers where it occurs. So we have the photosystem 2. I know it might not be clear here, but it's photosystem 2, or I could like try increasing it. Give me 1 second. Okay, I think it's the same thing. So when we're talking about any type of light dependent reaction, we have photosystem 2, and photosystem 1 mainly. Then we have the ATP synthase, which you should know about. Then we have the thylakoid, and all of this, remember, all of this occurs in a thylakoid. So what happens is, when you're receiving energy from the sun, when you're receiving energy from the sun, what happens is the energy is absorbed by photosystem 2. And it's weird that it starts with the name PS2 and not PS1. You have to like be very careful about this. Which starts first, which is the first photosystem, and it's photosystem 2. And it's because photosystem 1 was discovered first, that's why it got 1 in its name. Whereas this was discovered later on. So once sun hits the photosystem 2 with its light, it absorbs it, and this excites an electron, which moves down the electron transport chain, and it goes till photosystem 1. And while this is happening, some of the protons, which we which we deem as H plus ions, are getting pumped across the membrane. This is the membrane. This is a thylakoid membrane, so it's being pumped across the thylakoid membrane, and it's establishing an H plus ion concentration here. So over time, while the electron goes through the electron transport chain, the hydrogen is establishing a concentration gradient here where this is high and this is low. So now to get the clutter out of the way, because we already have enough, so the light, which is basically a photon, the elementary particle of light, photon excites a electron, and you may think where this electron comes from. Well, it comes from the splitting of water. So water splits into one oxygen and two protons and two electrons. So you have protons and you have electrons. Electrons that are going through and protons that are going across. And once this happens, what happens is the electron goes through the tunnel. You might think of this as a tunnel. It goes through the tunnel until it reaches photosystem 1, where it is excited once again. And what happens in photosystem 1 is NADPH and hydrogen produces NADPH, which we talked about before as an electron carrier, and this will be important in the light independent reaction in the Calvin cycle, or as we call it. So the electron is being excited, it goes through until it reaches NADP plus reductase, where it forms NADPH, and this basically accepts the electron plus a proton to form NADPH. And you may think, well, what was the need for pumping all the hydrogen protons this side? Well, this is so that the hydrogen protons funnel down this enzyme called ATP synthase. So ADP, which is adenosine diphosphate, is turned into ATP, which we learned in the previous video, as you may or may not remember. So this gives us a net yield of 2 ATP. So what happens next is we have we have the light independent reaction, and this is a very generalized explanation of the whole process. It's a very sophisticated and intricate process, so you'll have to learn about it in a bit more depth, but I'm glossing over it right now just to cover your basics and to remind you of what's happening. And if we're talking about the light dependent reaction, the input and outputs of is photons plus water gives us, and this is the this is the output. It's not directly giving us anything, but it's an input. And what we have as an output is we have NADPH, which is an electron carrier, we have O2, which is oxygen, and we have some adenosine triphosphate. And what happens in the light independent reaction is light independent reactions gets its inputs from the Calvin side, from the light dependent reaction. So what happens is it requires ATP to fixate carbon from the atmosphere, which is found in the presence of carbon dioxide. So as you may or may not remember, the formula of glucose is C6H12O6. So we have the H, we have the O, but we don't have the C. And the availability of C we get from carbon dioxide. And the fixing of carbon dioxide from the oxygen, fixing means like basically getting the carbon dioxide. So, and the fixing requires energy, and we produced ATP here, so that means we have energy now. So we have CO2, and some reactions happens with ATP and NADPH, which gives us a sugar. In most cases it is glucose, but it may vary. And this whole light independent process occurs in the stroma, which is like the empty space we talked about earlier. And just to clarify, ATP and NADPH is needed for carbon fixation. We need this for carbon fixation, and this is the carbon we're fixating. So we have the input and output table here. So you may just like go through it quickly or take a screenshot, whatever suits you. So now we'll move on. Well, moving on to respiration, we now have cellular respiration. And as you may or may not know, you should know because exams are approaching, that respiration are of two types: anaerobic and aerobic. Sorry for my writing, I'm forgetting how to write. But aerobic and anaerobic, and anaerobic means without O2, and this means aerobic means with O2. So we'll be mostly covering aerobic in this part, but we'll just go over anaerobic a bit. So aerobic occurs in 4 steps, one of which is glycolysis. And lysis means breaking. So we're breaking down glucose into pyruvates, and this occurs in multiple steps, but basically glucose forms pyruvic acid or pyruvic acid, and we have so pyruvate is a 3 carbon molecule, and this glucose, which is a 6 molecule, is broken down from the middle, and it is used to make 2 pyruvates. So before going next, we'll talk about what it also produces. It also produces 2 ATP per pyruvate, so you have 4 ATP, but it also uses 2 ATP in the process. So we have 4 ATP minus 2 ATP, which gives us 2 ATP as a net gain in ATP. It's basically like an investment. You may invest in a stock where it's here so that it may or may not go up top. So it's like an investment for the cell to invest 2 ATP for much more in gain. So what happens next is the formation of acetyl CoA, which is basically like a coenzyme and is needed in the Krebs cycle. So what happens here, I'll just give a quick rundown. So this pyruvate breaks down to give us 1 acetyl CoA, and acetyl CoA is a 2 carbon sugar, not carb sugar, it's a 2 carbon molecule. Now if you're keeping track 2 into 2, since it produces 2 acetyl CoA, you have 4 carbon molecules. But in glucose we had 6, so where did the remaining 2 go? Well, the remaining 2 are turned into carbon dioxide, and it is quickly getting rid of. So we basically have an input in this step. We basically have an input of 2 pyruvates plus 2 coenzyme through which acetyl CoA is made, plus 2 NAD+, which is basically an electron carrier, but it's not carrying electrons at this very moment. It will carry later on. So this gives us 2 acetyl CoA, and whoopsie, it gives us 2 acetyl CoA plus 2 CO2, 2 carbon dioxide plus 2 NADH. So these two are now carrying, they are now carrying 2 electrons. So now that we're done with the formation of an acetyl CoA, next we'll go on to the Krebs cycle. So what happens in the Krebs cycle is each acetyl CoA, one acetyl CoA gives us so let's rewind back a bit. So when we're talking about the Krebs cycle, it's actually a very complex process. But since it's a revision, I'll only going through over the few important parts. So Krebs cycle is needed to make more NADPH and FADH2, which are basically electron carriers, and some ATP. And this is important in the next step, which is oxidative phosphorylation. And I know the word sounds big, but it'll get easier. So what happens here is one acetyl CoA reacts with a molecule called oxalate. I may not get the pronunciation right. Oxalate. And what this does is it gives us citric acid. And citric acid, you may or may not know, is found in oranges and lemons. And this gives us plus 1 ATP plus 3 NADH plus 1 FADH2. So now if you go back to the video and find how many electron carriers have been produced, you will find that we have 6 NADH from the Krebs cycle, because we have 2 acetyl CoA. So this will be multiplied by 2 to give this. This will be multiplied by 2 to give this. And then we have 4 more. So 6 plus 2 plus 4 is equal to 12. So now we have 12 electrons carrier. Sorry, my throat is a bit sore, but now we get onto the juicy part. Now we have oxidative phosphorylation, the big boy who produces most of the electrons of the ATP, should I say. So what happens here is it looks like very tough, but it's pretty similar as photosynthesis, where it goes through a tunnel and then pumps protons, and protons come back, and then this happens, so on and so forth. But to make it simple, what we'll first start by doing is we'll start by thinking of this as the final destination. Think of it as a factory where all the coal is needed. So all the electron carriers dump their electron here. So what happens is these electrons basically go through all these systems, and at the end we have hydrogen and some electrons plus 1 water plus 1 molecule of oxygen to give us water. So here we may have 2 hydrogen protons plus 2 electrons at the end of the electron transport chain, and then these combine with 1 oxygen to form water molecule. And this is important because if water wasn't here to basically accept these electrons, the electron transport chain would never take place, because then these electrons would be out of place. So oxygen is important, but what basically happens is, while this electron is full of energy, it pumps hydrogen to the mitochondrial intermembrane space, and this basically pumps a lot, and by a lot I mean a lot of hydrogen protons, and this takes place in the mitochondrial matrix, in near the inner mitochondrial membrane. This happens in the inner mitochondrial membrane. So we have hydrogen protons being pumped from the mitochondrial matrix to the intermembrane space. So now what happens is, now there's a high concentration of hydrogen protons here and low, and these want to go here, but they can't, because first they're protons, and and charged particles can't get through easily, and two, they have no medium of transport yet. So now that we have the electron being accepted by oxygen and hydrogen to make water, we have that part out of the way. But what happens here is, these hydrogen protons go back through ATP synthase to to basically turn ADP into ATP, and this produces a lot of ATP. So just for reference, in the previous steps we had two in the glycolysis and two in the Krebs cycle. So guess how many how many ATPs are produced here? It's a lot. Once again, I'll tell you. So it's approximately 26, because when we're talking about biological systems, we can't be certain for any sort. Maybe some of these ATP synthase is not that efficient, or maybe so many NDPH wasn't produced, or maybe some problem happened. So we basically take a range. So the range I'll encourage you to use is 30 to 34 ATP, because it's right in the middle. And to close it out, we basically have this graph that you look at. So this is glycolysis. This happens in the cytoplasm of a cell. This needs glucose plus 2 ATP to produce pyruvate, 84 ATP, and NADH. And this may seem a bit hard, but this is basically the formation of acetyl CoA wala process. And this is the location is in the matrix of mitochondria, which means the inside, and the substrate needed are pyruvate, and this produces acetyl CoA, NADH, and CO2. And oxygen needed. Oxygen is needed in every step except for glycolysis. So glycolysis can occur without oxygen. So next we have the Krebs cycle, and in Krebs cycle this also occurs in the matrix of the mitochondria, and this requires acetyl CoA and oxaloacetic acid, and this produces oxaloacetic acid because it's basically like a catalyst, so it doesn't get changed, and it has NADH, it produces ATP, it produces this, and it requires oxygen. And finally we have the electron transport chain. So this happens in the inner membrane of the mitochondria, and the substrate needed are NADH, FADH, ADP, and phosphate, and these are required to make, and these ADP and ATP and ADP and phosphate group is required to make ATP, because if there's no ADP you can't make ATP, and the products of this is NADH, FAD, and ATP. So this is basically the whole summary of the whole process of respiration, and in this video we'll be going over anaerobic respiration, but it will be very brief. So what happens is, in anaerobic respiration is, glucose still turns into two pyruvates plus 2 NADH. and this produces 2 ATP, and this is basically all the energy it produces. So now these two pyruvate may produce ethanol if it's a yeast or some other cell. Ethanol, 2 ethanol plus, the quantity is basically 2 ethanol plus CO2 plus 2 NADH. Or it may produce lactic acid, specifically 2 lactic acid. Lactic acid plus 2 NADH. NAD plus, my bad. This is also plus, my bad. So this will be NADH plus, NAD plus, and 2 NAD plus. And if you've ever gone to the gym, you may feel like soreness or like a cramp after your first day, and that is because of the lactic acid buildup. Your muscles require energy, and so when you're going for the first time, your muscles may not be able to produce so much energy to keep up with your workout. So anaerobic respiration takes place in your muscles, and which produces 2 lactic acid, 2 NAD plus, and the lactic acid is what gives you that sensation of the tingling and so of soreness. And ethanol is used in alcohols, and yeast is also used in, I'll just verify, so this is in muscles and this is yeast. Yeast, CO2 is the, we use the production of CO2 in the making of bread. So if we're talking about a bread batter here, it rises up till here, and what makes the bread rise is the bubbles of CO2 which is produced inside. And ethanol is used in alcohol, as you all may know. So this is the complete lecture, complete overview on respiration and photosynthesis. So thank you guys. Stay tuned for our next video, which will be on cell communication. Thank you so much.